Hi, I'm Cassidy Heflin. I'm from Maryland and I'm a graduating senior who played lacrosse for 4 years at Piedmont College and majored in biology on the cell and molecular tract. I've been accepted into the physician assistant program at PCOM in Suwanee, Georgia. Fun fact, the week before spring break I had my gallbladder removed, and now I'm working on a sheep farm to make money before graduate school. Today, I'm going to talk about the ontogenetic shifts in diet related to eye size in the water snake genus Nerodia. In order to understand the idea behind ontogenetic shifts, I must highlight the meaning and importance of phenotypic traits. Phenotypic traits change based on the function they serve and the phylogenetic history. In other words, the traits that are inherited from your ancestors. Phenotypic traits are physical characteristics that an organism possesses, which can include things such as eye size, color of hair, body length, and height. For the purpose of this project, we focused on eye diameter and head length, which I'll explain more later on. Looking at traits in a phylogenetic context is extremely important because traits are inherited. To the left, we have a picture of a Nerodia water snake. Here, to the right, I have given an example of a phylogenetic tree, which shows the evolutionary history of animals. This tree demonstrates the relationships among the organisms, and we can make inferences about the evolutionary relatedness of mammals from this tree specifically. Traits are inherited, and we can use phylogenetic methods to understand the patterns of inheritance and traits of interest. However, sometimes inherited traits change as the organism goes through their stages of life. This is called an ontogenetic shift. Ontogenetic shifts can occur with morphological traits, including things such as eye size or dietary shifts, such as shifting from one type of food to another. An example of this includes photosynthetic foodstuffs, which are consumed by tadpoles. As tadpoles mature into adult frogs, they begin to consume insects. Ontogenetic dietary shifts are the development or change of feeding behaviors and patterns as an organism matures. For example, certain species within the North American water snakes, genus Nerodia, may experience an ontogenetic shift in diet from fishes to frogs. Specifically, as reported by Gibbons and Dorcas, both Nerodia fasciata and Nerodia erythrogaster eat small fishes, such as Gambusia affinis, which is pictured to the top left. And as the snake matures, their diet shifts to frogs, such as Rhena temporaris, as pictured on the bottom left. Nerodia rhombifer also shows an ontogenetic dietary shift, preying on small fishes as juveniles and shifting to larger fishes as adult snakes. The endemic North American water snake, genus Nerodia, consists of 9 described non-venomous snake species in the family Colubridae. All 9 Nerodia species are semi-aquatic, inhabiting both land and water, but they prefer aquatic habitats. To the right, there are pictures of a few of the species we use in our research. Typical habitat of Nerodia can be seen in the picture on the left. In other groups of snakes in the family Colubridae, it has been shown that eye size and visual acuity is tied to their environment. For example, arboreal snakes have larger eyes than either terrestrial or semi-aquatic colubrid snake species. Therefore, eye size may reflect habitat-specific adaptations and strategies related to predation. Some species of Nerodia feed on fish and others on frogs. Because eye size may reflect habitat-specific adaptations and strategies related to predation, we investigated if there was an ontogenetic shift of eye size and placement as related to dietary shifts. The various species of Nerodia had different sized eyes. For example, Nerodia erythrogaster, pictured on the top left, has among the largest eyes of any Nerodia species, whereas Nerodia cybodon, pictured to the top right, and Nerodia palsimaculata, pictured to the bottom left, appear to have smaller eyes. From these 3 examples, it is known that Nerodia erythrogaster experiences a shift in diet from fishes to frogs, and presumably capturing frogs requires high visual acuity and larger eyes than would be required for catching fish. Knowing this, we investigated the presence of an ontogenetic shift in eye size related to the proportion of frogs in the diet of 9 Nerodia species. Diet methods. For this, we obtain dietary information for each Nerodia species from the scientific literature. For each Nerodia species, we quantified the proportion of fish, frogs, and other prey items in the diet. These findings were used in subsequent analyses. Eye diameter and head length was measured for at least 24 specimens for each of the 9 Nerodia species. These specimens were obtained from the Natural History Museums housed at the University of Georgia, University of Texas El Paso, University of Texas Austin, and Auburn University. Some species included Nerodia cypodon, Nerodia cyclopean, and Nerodia rhombifer. For museum specimens, it is nearly impossible to stretch the snake out and obtain an accurate body length measurement. Therefore, we used head length as a surrogate for body length. We measured head length from the tip of the snout, represented by the red circle, to the location of the quadrate bone of the skull, which is represented by the blue dot. Next, using a computer program known as Fiji, we calculated the placement of the eyes using angles of converging lines in the rostral and dorsal view. The picture of the Nerodia erythrogaster, which is pictured on the left, is positioned in the rostral view. Using the lines from Fiji, we were able to create an angle starting from the bottom apex of the left eye, passing through the top apex, and then back down through the top apex, ending at the bottom apex of the right eye. This created the angle, and the angular value was calculated by Fiji and recorded. The next image of the Nerodia fasciata, which is pictured on the right, is positioned in the dorsal view, as if you were looking down at the back of the head. The angle was created, calculated, and recorded in a similar manner as the rostral angle, only using a different view. In StatView, morphological data were log transformed to linearize the data. An ANCOVA was used on the log transform data to test for differences in eye diameter among species. Head length was used as a covariant. The proportion of both fish and frogs within each species' diets were quantified from literature. As the graph shows, Nerodia fasciata has the second largest percentage of frogs in their diet at 10.80%, while N. erythrogaster has the largest proportion of frogs in their diet at 34.55%. This is interesting because both species have larger eyes than the other Nerodia species. Notice where the pink arrows are. Nerodia taxus pelota, Nerodia rhombifer, and Nerodia cyclopean have very small percentages of frogs within their diets. The results of the ANCOVA gave 3 p-values, all less than 0.0001, revealing a significant difference in eye diameter among Nerodia species. There was also a significant difference in head length among species, and a significant difference in slope, which is the relationship between head length and eye size. This is a graph of the slopes of lines regressing head length and eye diameter for the 9 Nerodia species. Of interest is the concavity component, which is the amount of curve present within the line. There was a large concavity in both the Nerodia erythrogaster, represented by the slope of the red dots, and the Nerodia fasciata, represented by the slope of the blue dots. We found a significant relationship between eye diameter and the proportion of frogs in the diet for these 2 Nerodia species. The graph to the top left represents a positive correlation between head size and eye diameter within Nerodia fasciata. Nerodia erythrogaster also represents a positive correlation, which can be visualized on the right graph. In both species, as head length increased, there was a marked increase in eye diameter. As shown in the bar graph, both Nerodia fasciata and Nerodia erythrogaster consume frogs when they are 50 centimeters or larger in length, as shown by the orange bars. However, small snakes, less than 50 centimeters, consume fewer frogs, as shown by the purple bars. These findings suggest that there is an ontogenetic shift in eye size related to the proportion of frogs in the diets of Nerodia fasciata and Nerodia erythogaster. On the other hand, Nerodia floridana, represented by the graph to the left, and Nerodia taxus pelota, represented by the graph to the right, have a negative slope for the relationship between head length and eye diameter. So, these species have small eyes compared to the size of their head. These findings in Nerodia floridana and Nerodia taxus pelota suggest that eye diameter does not increase as head length increases. Interestingly, the proportion of frogs in the diets of Nerodia floridana and Nerodia taxus pelota are extremely low compared to Nerodia fasciata and Nerodia erythogaster. These findings suggest that there is not an ontogenetic shift in eye size related to the proportion of frogs in the diets of both Nerodia floridana and Nerodia taxus pelota. We found an ontogenetic shift in the dietary proportion of frogs in Nerodia fasciata and Nerodia erythogaster that was correlated to an increase in eye diameter and head length. However, there was no evidence of an ontogenetic shift in the proportion of frogs in the diet related to increased eye diameter in Nerodia taxus pelota and Nerodia floridana. Ontogenetic shifts in the diets of water snakes may be related to changes in the jaw musculature and bite force that occur as the snake matures. Some species of Nerodia consume large frogs, which may require maximum bite force generated by the jaw musculature and associated cranial structures. In addition, larger Nerodia species, such as Nerodia fasciata, may experience an ontogenetic shift in diet from juveniles that consume fishes to adults which feed on large amphibians, such as frogs. Thank you to all of my professors in the Natural Science Department for helping me grow over the last 4 years. And thank you to all of my family and friends for the support and love. If anyone has any questions, please feel free to reach out to me at the given contacts. Thank you for listening.